want to forget that. So prep for Friday. All right, we're going to have a quiz in class, and that's going to be on the material that we have covered since the last quiz. So I believe that's last Friday up until today. All right, then don't forget you have that assignment 1 due on the 27th.
Oh yeah.
Don't wait till last minute because right after assignment 1 is due, we have the exam. Yeah.
Was open notes or no?
No, it is not open notes. Yes. So this is a little bit more high stakes. Remember, at the end of the semester, I am going to drop your 2 lowest scores. All right, so you have to miss an exam or you do really poorly on an exam, 2 of those scores will be dropped.
Do you happen to have a textbook update?
Textbook update. I do not. I know there's still, I think now there's 4 copies available in the library for you to either check out or look at it in the
We
library.
can check them out.
Potentially. I'm not sure.
I don't think we can.
No?
I think you can only use it in the library. That's what I thought.
Okay. Well, there are some in there for you to use.
So which of like the slides, like were from last Friday?
Oh, I forget what we talked about last Friday. You'll have to go back up on Moodle and look to see what it is.
Cell structure and organization.
Cell structure organization. All right, that is what we started talking about on Friday. Awesome, thank you. All right, any other questions pertaining to the quiz? Anything you should be expecting?
Is it just like lecture based, or should we be reading a book and?
It is lecture based. I think the book is helpful in giving you a little bit more background, if in case you just want to know more or you need to have it stated in a different way.
As far as like
What was that?
do you think that having not having a textbook would negatively impact the textbook? Or can I just look at the lecture?
Looking at the lecture is going to be fine. Yeah.
Great.
The thing about having the textbook is it does have extra questions to help quiz yourself, and it just kind of takes things in a different format in case you need that.
How many questions are in the quiz?
I want to say there's 10. I haven't finalized it yet. Yeah.
Is it multiple choice or like?
Very similar to what you had on Moodle last week. Like multiple choice, true or false. There might be like 1 on the blank. Yeah.
Do you ever use a case for the exam?
Yes. So study guide. I am going to make a study guide. I will post that tomorrow. I think study guides are a little redundant in the classes that I teach because it's basically just reiterating everything I want you to know off the lecture. All right. So study the lectures. You'll be great. Study guide is just kind of like a security blanket. All right. So anything on lectures, fair game. Anything that I've asked you to look at or read in preparation for a class, also fair game. Additional resources, I'm not going to test you on that.
If we were looking in the textbook, the exam next week, is that like just, I guess, like chapter 1 in the textbook, or is it?
If you look on the course schedule, for every day, it lists what chapters we would have covered. Yep. Yeah.
Wait, so I'm confused what you're saying. Like, you're saying, like, the things in the textbook are fair game too, or do you mean, like, the videos and stuff that
The videos that I've asked you to watch. So like the videos on the carbon atom, for example. Because we just reviewed it in class, but that went into it just a little bit more in depth. I don't expect you to memorize everything from those videos. I just want you to get those concepts. Any more questions about the exam? Study the lectures. You'll be fine. All right, remember quiz on Friday. Let's get started with today's lecture. All right, because there is a little bit to go through today. So we're going to start with cell membranes and transport. All right, I'm going to skip right to that first slide. All right, so cell membranes. We are talking about a phospholipid bilayer. All right, so 2 layers of phospholipid molecules. We know that phospholipid molecules have a polar region and a nonpolar region. And those nonpolar regions are going to be oriented towards each other. All right, that's what creates this bilayer. Now, when we're talking about the bilayer, we can kind of separate them out into, say, 2 leaflets. All right, so as a whole, it's a bilayer, but we can say we have 1 leaflet that faces the extracellular environment and 1 leaflet that faces in towards the middle of the cell. Now, an important note about these leaflets is that they're not symmetrical. All right, they're very much asymmetrical. All right, so some of the proteins that we might find on the outside of a cell, such as these glycoproteins, right, those are just proteins with carbohydrates attached. They're going to be more common on the outside of the cell, and they're really used for like cell recognition, right, so to help cells recognize each other. Hence, they're way more useful on the outside of the cell than the inside of the cell. Now we call this the fluid mosaic model, all right, because the cell membrane is not a rigid structure. All right, so the phospholipids and a lot of the proteins in this bilayer, they can move around. All right, so it's not a static thing. It's very much a fluid movement within the plasma membrane. All right, so I mentioned the phospholipids. We also have those membrane proteins I just alluded to. And we're going to talk about 3 of our plasma, of our membrane proteins. All right, the first one is a transmembrane protein. All right, they're going to be regions that are physically embedded into the hydrophobic region of the phospholipid bilayer. All right, so if we think about the hydrophobic region, which means water-fearing, all right, that's going to be in the middle of our membrane. All right, now regions on this protein have a section of amino acids, all right, that are also hydrophobic, and that allows them to be inserted into this region. All right, so we're physically inserting this protein. A lot of the times, this image is up here to show that a lot of the times that region that is hydrophobic in these proteins are going to consist of alpha helices. The second type of membrane protein is the lipid-anchored protein. All right, so this is a protein that has a lipid attached to it that's going to anchor it into the plasma membrane. All right, it's going to be covalently attached to our lipid, and that's going to anchor it in place. And then finally, we have our peripheral membrane proteins. All right, so these are proteins that are going to be non-covalently bound. All right, so if they're non-covalently bound, we're talking about hydrogen bonds or ionic bonds to either an integral membrane, all right, so one of these transmembrane proteins, or to a group of these polar heads of the phospholipids. All right, so in this image, it's bound to a transmembrane protein. It could also be bound just to a grouping of the phospholipids. All right, so lipid bilayer is fluid, all right? That's why we call it that fluid mosaic model. So molecules have the ability to move in our membranes. So when we're talking about phospholipids, all right, they have 2 planes of movement. They have lateral movement, so they're staying within the same leaflet of the membrane, but they're able to move around. But there's also rotational movement, where that phosphate head can essentially rotate. Even though the lipid is staying in place, it can rotate. So those are kind of our 2 dimensions of movement for phospholipids. Now, there is another movement that a phospholipid can undergo, and that is what we call the flip-flop movement. All right, super scientifically named there. But essentially, these phospholipids will go from one leaflet to another. All right, it's flip-flopping which leaflet it's on. Now, this is not energetically favored, all right, because it takes energy for a phospholipid to do this, and it needs this enzyme of flipase to facilitate this. All right, so first of all, we need an enzyme. Second of all, we need energy input. All right, so it's not favored. Now, the reason that they do this, and I actually had to go down a rabbit hole of scientific literature to try to figure this out, but essentially the phospholipids will need to flip-flop sometimes as the cell grows or as proteins start to rearrange, just to keep things kind of even on both sides of those leaflets. All right, so it's not favored, but it does have to happen sometimes. All right, so these membranes are semi-fluid. All right, so we have movement, but not everything is going to move. All right, so factors that might influence how fluid a membrane is, we have the length of the fatty acid tails, so how long these tails are on our phospholipids. Now, shorter tails, they're less likely to interact with other tails on the opposite side, so they're able to move around a little bit more freely. Makes it more fluid. So if we had longer tails, essentially they're going to become intertwined. It's going to inhibit movement a little bit. The presence of double bonds is also going to influence fluidity. All right, so if we have that double bond, it creates a kink. All right, and that makes it harder for the neighboring tails to interact. All right, so because it's not able to interact, it's more free. It has more fluid movement. Now we also have the presence of cholesterol. All right, so cholesterol tends to stabilize membranes. And the effects of cholesterol is going to vary based on the temperature. All right, so if we have high temperatures, our membranes become more fluid. If they're more fluid, all right, sometimes they start to get leaky. All right, we don't want leaky membranes. So what a cell can do is it can add in more cholesterol to help stabilize the membrane when it gets to higher temperatures. Now the opposite is true in colder temperatures. All right, so if we drop the temperature of a cell, that membrane is going to become less fluid. And if it's too solid of movement, it's not fluid, that's going to start to impact the function of some of the proteins in the membranes. All right, to try to inhibit this, we're going to take away some cholesterol. All right, make it a little less stable so it's more likely to be more fluid. Now, a cool thing about this is that a lot of cells can adapt to these different factors influencing fluidity. So animal cells, for example, will adapt based on the temperature. All right, so if it's too cold, they'll add more cholesterol to make it more fluid. Or excuse me, I said that opposite. If it's too cold, they're going to take away cholesterol to make it more fluid, to not have it as stabilized. And so they can adapt with the temperatures. Plant cells are pretty cool if they're adapted to live in hotter environments. Right, they're exposed to higher temperatures. They actually have membranes that have longer tails and fewer double bonds, right? So make sure it's a little bit more rigid and less fluid in those high temperatures. All right, so pretty cool how, evolutionarily this has evolved, and now animals and plants are able to take advantage of this. All right, so besides phospholipids, we know that we have transmembrane proteins, or membrane proteins in general, and these proteins can also move. All right, now not all of these proteins are going to move. Some of them are going to stay in place, but about 30 to 90% of these proteins can move depending on the cell type. All right, so there's a pretty cool experiment that was done that kind of shows how we know that proteins can move. So essentially, we took a mouse cell and a human cell, and we fused them together. All right, now the mouse cell has this protein, this H2 mouse protein, that was bound to a fluorescent dye. All right, so it's bound to this fluorescent dye, so we have the ability to look at it under a microscope. Now at cold temperatures, we know that the membranes are less fluid. They're not going to move as much. And so under cold temperatures, they saw that the fusion, you can see exactly where that divide happened. All right, this half is mouse, this half is human. But when they warmed up the membranes, right, made it more fluid, all right, and then they took a look at the cell again, they saw that some of the mouse proteins had moved within the membrane of that hybrid cell. All right, so they were able to see that these proteins were physically moving, and hence we know that proteins can move in the plasma membrane. All right, now like I said, not all proteins are going to move. All right, so there are going to be some that are bound to structures inside or outside of the cell, and those are not going to be moving at all. So they can be bound to like a cytoskeletal filament. Looks like this is probably an actin filament here. All right, it can be bound to the cytoskeleton. It can also bind to the extracellular matrix. All right, so outside of that plasma membrane. So if they're bound, they're staying put, they're not moving. All right, so we talked a little bit about the components of a phospholipid bilayer, but one of the key things that this bilayer does is that it provides a barrier. All right, so it's a barrier to hydrophilic molecules and ions due to the hydrophobic interior. All right, so because hydrophilic molecules are water-loving, they don't want to go into this water-fearing region in the middle of our bilayer. All right, so it prevents a it presents a barrier. And that's going to depend on a few things, all right, depending on the molecule we're looking at. So size, smaller molecules tend to diffuse faster. All right, so we see up here nitrogen, oxygen, carbon dioxide, they're diffusing pretty easily. All right, so it has a high permeability to these molecules. Polarity is also going to influence how fast it's going to diffuse across the plasma membrane. So non-polar molecules diffuse faster, again, because they're water-fearing if they're nonpolar, so they can go through this middle of the plasma membrane pretty easily. And finally, charge. All right, so non-charged molecules can diffuse fast as well. Same basic concept. They're non-charged, they're hydrophobic, they can pass through the middle of this bilayer pretty easily. All right, now these factors are all going to combine together depending on what molecule we're talking about, and they're all going to influence whether or not a certain molecule can pass through this barrier. All right, so passing through this barrier, there's several different ways that this can happen. All right, so one of the ways is just simple diffusion. All right, it's passive transport. Molecules can move from one side of the plasma membrane to the other, and they follow a concentration gradient. So they're going from areas of high concentration to areas of low concentration. All right, this is typically going to be smaller molecules that will be able to do this. The next type is facilitated diffusion. All right, so facilitated diffusion, also passive transport. We don't need any energy associated with this. All right, we're also going from an area of high concentration to low concentration, but it's being facilitated by a protein. All right, and in this image here, we have a channel protein that just allows these solutes to move from one side to the other a little easier. All right, but we're still following a concentration gradient. Now the difference between passive transport and active transport, which is our next type of transport we're going to talk about, is that active transport needs energy. And it needs energy because now we're going against a concentration gradient. All right, so we're going from areas that are already low in concentration to areas that are high. All right, so we're going against the gradient. So passive transport requires no energy. All right, particles are going to diffuse down their concentration gradient and eventually reach an equilibrium. All right, so we can see in this image here, all right, one side definitely has more molecules than the other. All right, so they're going down their concentration gradient onto the other side of this membrane until eventually they've equalized. All right, so no energy involved, pretty simple. This can happen with multiple molecules at the same time until we reach some sort of equilibrium. Now, even though we are at equilibrium, that does not mean diffusion has just stopped. All right, diffusion is still going to occur, but there's not directionality to it. All right, so instead of saying in this first image that all these molecules are going to diffuse in a certain direction across the membrane, all right, at this point there's still diffusion, but they're not going one way versus the other more than each other. Does that make sense? All right, yes.
So like the back and forth is equal to one another.
Yes, that's a way better way of putting it. The back and forth are equal to one another. All right. So we have more terminology when we're talking about solutes and gradients. And this is all based on what solution you're talking about and what side of the membrane it is on. So if we're talking about an isotonic solution, right, we're talking about a concentration inside of a membrane and outside of the membrane that are the same. All right, so isotonic, those concentrations on either side are the same. Now that's different from hypertonic, right? So hypertonic is referring to a solution that has a higher concentration or more solutes on one side of the membrane than the other. All right, totally opposite of that is hypotonic. Hypotonic is a lower concentration. All right, so depending on what side of the membrane we're talking about here is going to determine how we're talking about these solutions. All right, so let's take this middle image, for example, and it says the outside is hypertonic. Yes, that's definitely true, has a higher concentration of these molecules. right? But this inside would be hypotonic, right? Because it has a lower concentration. But now if diffusion has happened or maybe some equilibrium has been reached, something's going on here to make the outside of the cell now has become hypotonic, lower in concentration. All right, so when we're talking about these terms, it's all in reference to a membrane. All right, so we're talking about reaching equilibrium. So cells need to maintain gradients. All right, so they are maintaining an internal environment very different from their external environment. So they have to maintain a transmembrane gradient. And this is where the gradient of a solute is higher on one side of the membrane than the other. All right, so in this top image here, we're looking at our glucose molecules. And we can see that glucose is definitely in higher concentration outside of the cell. All right. We also can talk about an ion electrochemical gradient. All right, so when we're talking about this gradient, we're actually talking about two things working at the same time. The first is the charges. So irrespective of the molecule, we're just looking at the charges, and the charge represents an electrical gradient. So if we look at this cell, inside of the cell has more negative charges, so it's going to be more negative relative to the outside of the cell. All right, but it also has another component in that we're also looking at the chemical gradient. All right, now we're looking at the specific solutes itself. So if we look at chlorine here, we see that's in higher concentration inside of the cell than outside of the cell. Now, one thing to note with these gradients is even though there's a huge difference between the internal and the external environment, for that cell, that could be equilibrium. All right, so equilibrium doesn't have to mean that there's equal concentrations on either side. Equilibrium can mean that the concentrations are what they need to be on the inside versus the outside. All right, so what if we're talking about solutes that have a hard time moving across a membrane? All right, when we're talking about these solutes, this is where osmosis plays a huge role in creating a type of equilibrium. All right, so osmosis is the diffusion of water across a selectively permeable membrane. So if the solutes can't move, the water will. So in this image here, we have our purple solute molecule, higher concentration on one side than the other. All right, the solute can't move, but that water is going to move to the other side to try to balance things out. All right, so it's trying to get that ratio of solutes to water molecule equal on either side of that membrane. All right, this bottom image kind of shows you how this is happening in a little bit more detail. So essentially you can think of it as the solute molecules are attracting the water molecules across this membrane. And because we know water is polar, all right, they'll be attracted to these molecules if they have some sort of charge to them. All right, that's a little caveat there. So water balance, super critical. Yes.
Can you go to the last slide?
Yes, I can.
So, like, when you say that their osmosis is occurring, like they're trying to get the same, like almost molarity, like the same number of
Yes,
on
exactly.
each side.
Yep. Any other questions? All right, so this water balance is really critical if we look at whole organisms or individual cells. All right, so tonicity is the ability of a solution to cause a cell to gain or lose water. So basically what we're talking about is the cell hypertonic or hypotonic. This is really important in osmoregulation, which we're not really going to go into in this class, I don't believe. But that's the control of water balance within an organism. All right, and fish, this can be super critical. All right, so we have our marine fish here. Marine fish, they live in salt water. All right, so because the solution around them is hypertonic, all right, there's a ton of salt molecules around them, water inside of that organism is constantly going to be wanting to exit. All right, so they have to basically fight to keep their water in. Totally opposite if we're talking about freshwater fish, all right? Now the water around a freshwater fish is more than likely hypotonic, so lower in a concentration of solutes. All right, so they have a problem of keeping the water out because the water constantly wants to enter in to try to get the molarity the same, create that equilibrium. All right, but the equilibrium for that fish is not the same as what the equilibrium for the water wants it to be. Now, in individual cells, if we look at a smaller scale, also presents a problem. So if we look at animal cells, all right, this is red blood cell. If we place it in a hypertonic solution, all right, so lots of solutes outside of the cell, water is going to want to leave that cell, and basically that cell can implode. All right, it's going to collapse on itself. Opposite is true if we place it in a hypotonic solution. All right, so now there's more solutes inside of the cell. Water wants to rush in. But the problem with that is that these cells can swell and eventually they can rupture. All right, so very important in maintaining that homeostasis, maintaining that important water gradient inside and outside of the cell membrane. Now, plant cells are a little different, but same basic concept. So plant cells, they have that rigid cell wall around them, all right, and that rigid cell wall keeps their structure. So if we place it in a hypertonic solution and water wants to leave the cell, right, the cell doesn't shrink itself, but the plasma membrane will shrink away from the cell wall. And that could cause that to break apart and implode as well. Right, so we're shrinking away from the cell wall. The cell wall doesn't change because it's a very rigid structure. Now if we place it in a hypotonic solution, water is going to rush in. Right, it's more than likely going to fill up this large vacuole here. Right, and it's going to cause that cell to swell, but it's not going to swell to the point of bursting because that cell wall is keeping that water in place. All right, so cell wall plays an important role in plants. All right, so group question here. I want you to think about the different types of transport and discuss the advantages and disadvantages of simple diffusion, facilitated diffusion, and active transport. And as you do that, I'm going to come around and hand back your H2O assignment. So we're talking about a hypertonic solution.
Oh.
Water is out. Does that make sense?
I have a presentation 10 minutes after this class.
So that is, so we placed it in a totally different stats right? So now relative to the outside of the environment, there's way more solid inside of it. Now water was there. So we're only talking about water. What was that? Yeah, try to give now in real life, osmosis and all that transport, they're all gonna work together. So a little bit of both, but if we just look at one or the other, that's all we want.
Oh.
Looks like a guy with 2 eyes.
All right, has everybody had a chance to work through the question? Nice. I know I did not pass all these back yet because I am still learning all of your names. I will continue to pass those back in a moment here. So what might be the advantage of simple diffusion? Yeah?
No energy is needed.
No energy is needed. Yes. What might be another advantage?
Transport proteins are needed either.
Don't need any transport proteins. Anything else? Okay, it can be very fast. All right, what about a disadvantage?
It doesn't really work for anything that's bigger and has charge.
Yeah, it doesn't work very well for charged or larger molecules. Any other disadvantage we can think of? Yeah?
Sometimes the cell needs to have a lower or higher than the outside environment.
Yeah, so sometimes the cell wants to regulate it a little bit better, and there's no point of regulation. Exactly. All right, what about facilitated diffusion? Right, what's an advantage? We can allow larger molecules that can't pass through the phospholipid bilayer. Yep, maybe they're hydrophobic. Any other advantage?
I believe no energy is needed for that one either.
No energy is needed. Yep. Anything else? All right. I'm going to throw in something that we really haven't talked about quite yet, but sometimes those proteins can be specific to a certain solute. So maybe it's helping transport in something that the cell needs. What might be a disadvantage to facilitated diffusion?
You still aren't able to get the biggest of molecules through, like
Still can't get the biggest of the molecules. What might be another disadvantage? It requires a protein. Yeah, protein's energetically expensive to make. Anything else? All right, I'm going to say exactly what we said for simple diffusion is that typically there's not as much of a barrier as there would be to other types of transport, such as active transport. All right, what might be the advantage of active transport? Yeah?
You can go the other way, like not parallel.
Yeah, you can go against a concentration gradient. What's another advantage? Yes?
you can have like ions and other polar molecules move through.
Yeah, you can move through ions that might not be able to get through either by simple or the facilitated diffusion. Any other advantage we can think of? I think you covered the main ones here. What might be a disadvantage?
You need energy and ATP for it.
You need energy. That's the huge disadvantage. What's another disadvantage? You need a transport protein. Any other disadvantage we can think of?
It's easier to mess up, like a disease or something, where to mess that up is a lot easier. Simple, at least.
All right, I can see that. Yes, yes. So I'm not too up to scale on diseases and how they work with transmembrane proteins, but because these membrane proteins are specific to something, if a disease comes in, it could wipe out something that's specific to it. Generally speaking here. All right, any questions about what we've talked about so far? All right, I have like 5 minutes, so we're going to do what we can here. 4 minutes, actually. All right, so transport proteins. All right, transport proteins are going to provide a passageway for the movement of ions and hydrophilic molecules across the membrane. All right, so we kind of talked about transport proteins already with facilitated diffusion and active transport. But there's two basic types of these proteins, and that would be a channel or a transporter. So channels, they form an open passageway, right? Open passageway for ions or molecules to move across a membrane. Now, most of them are going to be gated, meaning we can close that passage. All right, so I like to think of this as a door in the wall. All right, you open the door, we can move solutes in. But we can also close that door and stop anybody from passing through, right? How that's stopped is normally by a type of protein that's going to bind to this protein and just cause that gate to close, all right? We're not going to go into specifics of that. Ligands are one of those that'll do that. I might have just misspoke, actually. I'm not sure if a ligand is a protein. I might have to get back to you on that. But ligand is the molecule that would bind and cause that to close. The good example of a channel is an aquaporin. Right? An aquaporin, we can find them in our kidney cells as well as a lot of plant cells, especially in the roots. They're really important for helping speed the movement of water across the membrane. Right? So they are very specific to water molecules. The other type of transporter, or the other type of protein is a transporter. They're also known as carriers, though they don't physically carry anything across. Right? And this is where the protein goes through some sort of shape change. Right? So this is going to be the principal pathway for a lot of the larger molecules or charged molecules such as sugars, amino acids, and nucleotides. All right, so there's basically two features to these transporters that help facilitate movement. All right. One is going to be a hydrophilic pocket. All right, so the hydrophilic pocket, that's where these solutes are going to enter into the transporter. And the other big thing about these membranes is that they can change their structure. All right, they're able to switch back and forth between two conformations. So we have our solute coming in, it's changing its shape, and allowing that solute to exit on the other side. All right, so 3 types of transporters. I think we'll get through this slide and then we'll call it quits here. So the uniport transporter, or uniporter, single molecule or ion. All right, just a single ion or molecule. It's going in one direction. Symporter, on the other hand, still going in one direction, but it can transport 2 or more molecules. And finally, we have an antiporter. All right, 2 or more ions can be transported, but they can be transported in opposite directions. All right, any questions about what I have talked about so far? All right, we're going to pick up on that slide on Friday.